This is the core component mock, and this is paper 2. And we're on question 2, and it shows, diagram shows epithelium found in a mammal. So a mammal like you, epithelium is a lining of something. State the type of microscope used to view the epithelium preparation. So a few clues here. One is times by 1,000, and you might have actually used 1,000 in class. Our microscopes at college actually go up to 1,000 with the right lenses fitted. It's slightly blurry at that magnification. So that's sort of the limit of light microscopy. So this kind of fairly blurry image at 1,000, that's going to be a light microscope, so not an electron microscope. So light microscope or optical microscope would do there as well. Name the parts labelled X and Y. One of these would be okay, the other one might be a little bit more difficult. X. So if I draw another sort of cell there with the nucleus, that's kind of Bart Simpson kind of haircut. And we're looking at this being a brush border or microvilli type thing that you're looking at, but these are not actually microvilli like the digestive. They're actually cilia that can beat and move dirt. So dirt and bacteria, they can beat and move these along. So brush border, so it looks like a brush, and they're actually cilia that we're looking at there. So cilia or a brush border. Y is a kind of membrane underneath that all of these cells are attached to, and that's a basement membrane. So maybe those two might be slightly tougher to answer. So such cells are found in the trachea, so the windpipe. Explain how the structure of the cells is related to its function. So, as we said before, the bacteria dust particles can be moved along with mucus to keep the linings of these very important tubes free of dirt and blockages. So the cilia move, beat, or that sort of, so move or beat, or in waves of movement, and the mucus, with dust, bacteria, or whatever's there is carried along, and eventually you cough it up or you swallow it, and that's how you get rid of that mucus. Two marks for calculation. You should be fine with calculations. You've done the skills homeworks. Calculate the actual height of the cell shown between A and B. Show your working. So A and B is there. So what we've got to work on, we've got a ruler, and so we can measure a distance there, and we've got the magnification. So on the page that is, on my page I'd go for 34. So, and I'm going to write down image size A to B equals 34 millimeters. Show your working. I'm not just going to write 34. I've told you what it is. And it's not really 34 millimeters because it's 1,000 times magnification, so it's 1,000 times smaller than that. So actual size equals 34 millimeters divided by 1000. So calculator, 34 divided by 1000 equals 0.034 millimeters. It doesn't ask for you to give specific units, so that millimeters is fine. If you're determined to give that in micrometers, then you would move your decimal place 3 places that way, and that would be 34 micrometers. So either of those are fine, but why bother changing it further and risk a calculator error? And the last bit, part E, just a definition for 2 marks. What is meant by a tissue? And you can just say it's a large number of cells with a similar structure carrying out a similar function, and that'll just get you 2 marks, nice and easy. So collection of many cells with similar structure carrying out a particular function, and this is the function of being an epithelium lining. So some straightforward marks. Calculation you've done many times before. You should have got a reasonable score out of 9 hopefully.